welcome to this 12th lecture in this academic year, and also the 99th lecture in the series in the engineering science lecture series since the start in 2000. Yeah, you can count it, in 2006. Also, I would like to thank Agilent Technology, now Keysight Technologies, for sponsoring this lecture series since the start in 2006. Before I introduce our guest speaker for today, two things. First, the pixel will be here at 5:30 right after the lecture, and you can hopefully enjoy it. And then also, I'm going to send the slide for, the flyers for the talk on September the 3rd, when the time comes in, probably in August timeframe or so. Our guest speaker for today are Dr. Nancy Reed, and then also Dr. Todd Reed, and the title of their talk is Augmented Phonocardiogram Acquisition and Analysis. Dr. Nancy Reed received the Bachelor of Science in Biology and Master of Science and PhD degrees in Computer Science from University of Minnesota in 1988 and 95 respectively. From 1993, Nancy lectured at Sonoma State University and was an adjunct assistant professor at UC Davis. In 1998, she joined the computer science department at Linköping. Okay, University in Sweden, and was awarded the docent in 2003. She joined the University of Hawaii in 2002 as an assistant professor in electrical engineering. She transferred to the Information and Computer Science department in 2004 and has been an associate professor in ICS since then. In 2008, she was a visiting scholar at UC Berkeley, and then she is, of course, also a member of a number of professional organizations, including IEEE and the ACM, and also the IEEE Computer Society. As far as Dr. Todd Reed, he received his Bachelor of Science and Master of Science and PhD degrees in Electrical Engineering from University of Minnesota in 1977, 86, and 88, respectively. From 1977 to 83, he was an electrical engineer at IBM, and from 1984 to 86, he was a senior design engineer for Astrocom Corporation, St. Paul, Minnesota. He served as a consultant to MIT Lincoln Laboratory from 1986 to 88. From 1989 to 91, he was the head of the Image Sequence Processing Research Group in the Signal Processing Laboratory, Department of Electrical Engineering at Swiss Federal Institute of Technology at Lausanne. From 1991 to 2001, he was on the faculty of the Department of Electrical and Computer Engineering at UC Davis. In 2002, he joined the Department of Electrical Engineering at University of Hawaii, Manoa, where he is currently a professor. In 2008, he was a visiting scholar in the Department of Electrical Engineering and Computer Science at UC Berkeley. He joined the graduate faculty in neuroscience, John Burns School of Medicine, University at Hawaii at Manoa, 2010. They are both here as visiting professor. In fact, Nancy is a visiting professor at Computer Science Department, and then Todd is a visiting professor here in this department. In fact, I think a number of you are, in fact, already in his class in image processing. So let me first introduce Nancy.
Okay, thanks everybody for being here. It's my pleasure to talk to you today about augmented phonocardiogram analysis, and don't worry, I'll define what that actually means shortly. So phonocardiograms is just a long drawn-out name for heart sounds. And so in the old days, when the phonocardiogram name was coined, they were actually plots on strip charts. And so, a piece of paper would fly by a pen and you'd see scribbles on the paper. And so that's where the gram comes from. These days, phonocardiograms are actually digitally recorded heart sounds and stored, which for people like us that do things like signal processing, that's good news, because doing signal processing on a strip of paper is kind of hard. But doing signal processing on a digital file, not so much. So if we're going to analyze these heart sounds, there are two main parts that we have to do right up front. The first one that I'm referring to here as, oops, my pointer went out. This other one, this one's.
Oh yeah, there we go.
Sorry. Okay, so the. It works for you, it doesn't work for me. Okay, well, I'll just point. So the first thing that we need to do, technical difficulties. So the first thing we knew that I'm referring to as detection of the various sounds produced by the heart, actually that's acquisition in our case. It's not enough to say, oh, there's a heart sound there, because that's kind of like saying, well, the patient's alive. Well, we kind of hope to know that. But what we really want to do is record the various sounds produced by the heart.
Are you using a stethoscope?
Well, that's a good question, actually. And by the way, that brings it up. Any questions, anytime. I mean, you don't have to save your questions to the end. If something comes up, ask it, because it's a lot easier for me to address it without having to think back several slides. So the good question is, how do you do that? One way would be, in fact, a stethoscope, like this. And these things are relatively inexpensive. They're really widely available. And they usually have a sound output of some sort, either analog or digital, that you can then record. And so for certain purposes, something like this is fine. I mean, if you're looking at one location and you want to acquire a sound at just that location, then this works beautifully. And then if you want to look at another location, which is what cardiologists do, you move on to the next location and you can record that sound, and that's fine too. There are some limitations to that, though, that I'll talk about in a little bit. So at first blush, it seems like, well, this is a solved problem, because you can buy these for $100 out in the snow. But there are limitations to them that I'll talk about. Okay, the second thing that we need to do is to remove or somehow ignore sounds caused by other factors. Now, in our case, we're working with babies. We're working with a pediatric cardiologist, and all of our subjects are either from neonate to about a year old. And so, for example, patient motion obviously is a big deal with kids. The baby's not going to sit there still while you try to put stuff on it and listen to its heart. And so this patient motion induces sounds into the stethoscope or whatever your sensor is or sets of sensors are. In our case, we actually hear the sensors to the patient, but even so, motion causes artifacts, sound artifacts. It turns out that the lungs are a very strong acoustic source. And we don't think of it, but when you see these doctors with the stethoscopes around their necks, actually what most of them are going to do is listen to your lungs, because they're listening for congestion and things like that. And so lung sounds can be a big noise source when you're trying to listen to the heart instead of to the lungs, because acoustically they get mixed because they're so close together. It turns out, again, because we're dealing with babies, that speech enters the problem. And the reason is not because the kid talks, because up to a year old they don't really talk much. They may cry a little bit, but they don't talk much. But always when you're dealing with a child of that age, the parent needs to be there. And fairly often the parent finds it impossible to not talk when you're trying to acquire the heart sound. And I mean, speech in the air is an acoustic signature too, and even though stethoscopes are relatively omnidirectional, they can't reject everything. And so for many of the heart sound recordings that we acquired, in fact, I can hear the mother speaking in the room. And so that kind of thing we don't want in our analysis. It really confounds things. And so you need somehow to deal with, like I say, either ignoring, which is pretty hard, or removing those types of factors. So it turns out, I mentioned all the stethoscopes around people's necks and that they use them for lung sounds. I had always assumed that if a physician has a stethoscope around his or her neck, that they know how to use it. And so when they put it up against my chest to listen to my heart, that they can tell what it is that's going on with my heart. Not true. It turns out that very few, especially general physicians and general healthcare workers, they can't tell whether your heart's working well or not. They can tell that it's beating. Good. But if it's diseased, they're unable to tell that it's diseased. They can't tell a normal from an abnormal heart sound. And even more so, surprising to me, was that neither can cardiologists. And so we're working with a really good cardiologist at Triple Army Medical Center, and he takes interns and crops and crops and crops of interns. And every single crop he has, he tries to train them to be able to make diagnoses based on what they hear through a stethoscope. And every crop, because we worked with him for 5 years or something, every crop he complains. Because not only can they not do it when they come in, he can't train them to do it either. It turns out to be a very, very difficult thing to teach people. It's kind of like trying to teach somebody perfect pitch. It's not so very teachable. And in fact, there have been formal studies that have shown that taking a cadre of interns and putting them through a training program, evaluating their skills before and after, their skills after are no better than their skills before. For all of the known training techniques. And so this is a pretty big issue, as you can guess. And so our goal is to try to provide, first of all, a mechanism to provide diagnostic advice, but also to provide a training mechanism. Because the thing that's missing with training people to hear these things is some kind of feedback. Because you can show somebody or have somebody listen to a sound and say, okay, there's a mitral valve click in that sound. Well, telling them that it's there, they can listen all week long, and if they can't hear it, they can't hear it. And so if you can have some sort of repetitive feedback that says, okay, here's a sound, what do you think it is? Here's the answer. And then can do that over and over again, there might be a chance. I mean, this hasn't been proven, but there might be a chance that that's an effective training mechanism where the current ones clearly are not. So, as I mentioned, this stethoscope, they're really widely available. And so previous work that has been around has focused on sounds taken from a single site. Site may be different sites, but it's a single one. Okay? And so I'll show you what sites mean in a second. But for example, there's a site called the aortic site that's right about here. And that's a very common one for studies in automated analysis of heart sounds. Also, it's very commonly the case, almost always the case, that in the published work, and by the way, by published work, we've now surveyed over 700 papers published since 1995. And so the vast majority of these things use single sites. By vast majority, I mean out of over 700, there are 3 that use more than one site. Okay. So, and under controlled conditions. Now, cardiologists don't work that way. Now, if we wanted to build a toy system that we would run in the lab and publish some papers on and then go on and do something else, okay. But our goal is to try to provide something that's actually useful And that's why we're working with an actual cardiologist, is to say, okay, is this going to be useful to you or not, and what do we need to do to change it to make it useful? Okay. So we'd like to be able to, in some sense, reflect the way cardiologists perform auscultation. Auscultation is listening to heart sounds. And so what cardiologists do is they use different locations. And the reason they do that is because different pathologies are most obvious sound-wise at different locations. So for example, if you have a mitral valve problem, then the location that's closest to the mitral valve is the one that you want to listen to detect whether that problem is there or not. Because if you're listening on the other side of the chest, the sound is attenuated. Whatever the effect is much harder to hear. And so what cardiologists do is listen in a bunch of different places, and I'll show you where they are. There are some standard locations that they look at. So what we're working with is working to use simultaneously acquired multisite phonocardiograms that are acquired under clinical conditions. So for example, if mom's in the room talking, our recordings show that. Yeah.
Do these work along with EKGs, or is it a completely different type of disease you're looking at?
It's similar diseases. They can be considered complementary. One of the reasons that phonocardiograms are still interesting, although EKGs are much more common right now, partially because they're a little easier to interpret and the equipment's become relatively inexpensive. But the problem is that EKGs, as the name implies, are tied to electrical activity. Because when the heart beats, for example, there's a wave of electricity, of electromagnetic energy that's radiated from that. That's what EKGs pick up. But there are a lot of pathologies that don't have electrical signatures. So, for example, if you have valve problems, that doesn't have an electrical signature associated with it, but it does have an acoustic signature. And so for also heart murmurs and things like that, they're not reflected in EKGs. Yep.
Sorry, are there any frequencies of interest in terms of acoustics of it that falls outside of the normal human hearing in terms of?
No.
So this is all inside.
This is all well inside.
20 hertz.
Yeah. I mean, the base frequency is the heartbeat frequency, so it's 70 plus or minus.
Oh, within the sound itself.
Oh, yeah, the repetition, the periodicity is around 70 or so. Within the sound itself, it's still under, say, 2 kilohertz.
Okay.
So it's not that the stuff is beyond human hearing. That would explain a lot if it was, but it's not.
Okay.
Other questions? Did I answer your question? Okay. So the other kind of implied issue here is that I said simultaneously acquired. And you may say, well, how come? Because cardiologists don't do that. They go from this site to this site to this site to this site, sequentially. And our goal was to examine, well, if we do that, how does it work? How well does it work? If we have each of these samples more or less in isolation, how well do we do? On the other hand, if we have them all time aligned, so that we can look across the different locations as well as along the locations, so that we can correlate things that we hear at the different locations all together, can we learn things that otherwise cardiologists don't learn? Now, when I first started this stuff, I thought, okay, well, I'm going to take a bunch of multiple locations. I need them time aligned. So I'm going to use this stethoscope. I'm going to get the signals, and I'm going to time align. And it turned out, because you could use, say, correlation or something, presumably, to do this. It turns out that heart sounds are sufficiently non-stationary. That is, their characteristics vary with time a lot. And so if you take, even at the same site, samples at 2 different times, because when you inhale and exhale, your heart sound changes. So if you just take 2 samples at the same site and try to correlate those, try to time align them, you can't. And I had a student work on that for 6 months. You just can't. The correlation function is just really flat. There's no discernible peak. It just doesn't work well. And what we'll be showing you is that not only do we want to time align things, but but we want a timeline between different positions. And the signals, as I'll show you, at the different positions are really, really different. So here's what we're looking at in terms of the structure. This is a very simplified schematic of the heart structure, but it covers the things that we're going to be interested in this talk. So we're interested primarily in the left and right atrium, the left and right ventricle, and also the valves that go between them. So there's the tricuspid, the pulmonary, the aortic, and the mitral valve. And these are important because they operate to produce the sounds that we'll analyze by closing in pairs. And I'll talk a little bit about the characteristics of how they close. Okay, so that sets the thing up. I'm going to have to move along since I'm eating up a lot of time. So what we're talking about here is referred to generally as the cardiac cycle. And what that means is the events related to the flow of blood that occurs from the beginning of one heartbeat to the beginning of the next. So when we talk about a heartbeat, we're talking about two things. One is called systole, which is ventricular contraction. Those guys that I showed you at the bottom there, they contract and squeeze blood out into the body. And diastole, which is ventricular relaxation, where they relax and pull blood into the atrium. Okay? So the sounds that are referred to as S1 and S2, those are components of the heart sound, and I'll show you more specifically what that means, mark the beginning of systole for S1 and the beginning of diastole for S2. So S1 and S2 are, when you listen to a heart sound, you hear bum, 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 bum. S1 is the bump, S2 is the bump. And so I'll show you a plot of that to make it a little bit more specific. But those are the two components that we're talking about so far. So here's a plot of a heart sound. And so when you first look at these, it's kind of like, what? Here's an S1, here's an S2, here's an S1, here's an S2, here's an S1, here's an S2. Here's a typical noise component that you need to do something about if you're going to work with this kind of signal. Now, how do I know that which one is S1 and which one is S2? Under normal conditions, the time spacing between the S1 and S2 is shorter than the time spacing between the S2 and the next S1. So under normal conditions, what are abnormal conditions? Somebody's under a lot of stress. They're running really fast, their heart rate goes up. When that happens, the difference between this and this becomes small. So under the normal conditions, you can use timing to discriminate between S1 and S2. Under abnormal conditions, you cannot. Okay, so the sounds that we've talked about so far are the S1 and S2, these guys. When it says normally present, that means if the person's alive. Okay, and so the S1 is due to the closure of the mitral and tricuspid valves that I showed you. The S2 is due to the closure of the aortic and pulmonary valves. There are other sounds referred to as S3 and S4. Those are, in normal people, very rarely present. Actually, generally speaking, they're rarely present. If they exist, they almost always indicate pathology of some sort. Another sound that wasn't shown in that previous slide are referred to as murmurs. They're basically sounds due to turbulent flow of the blood. And depending upon when they occur in the heart cycle, they get different names. So if you have one that happens during systole, it's called systolic. During diastole, diastolic. But you may have a murmur that completely covers the signal from beginning to end, in which case it's referred to as continuous. Another heart sound that you may encounter, or a component of a PCG that you may encounter, are clicks and snaps. Under normal circumstances, those aren't present. What they usually correspond to, if they are defects in valves. And so, for example, the valve closes and it doesn't close well. There's a condition, for example, called mitral valve prolapse, where the valve closes but it doesn't close properly. And so it makes a sound instead of closing like when you close a door on a good car, it just goes boom. No, this one's not clicking like a good door. It just clicks. The other is the case where people have artificial valves and the valves start to fail. And those also exhibit these clicks and snaps. Okay, so these sounds S1 and S2 I've indicated as kind of single components. But there are conditions under which they can be perceived as two components. Usually the S1 is described as or seen as a single component or heard as a single component because the tricuspid and mitral valves close very, very close to the same time. And as you breathe in and out, that doesn't change. In the S2, the valves close at slightly different times. It's referred to as a split. And the split between two different components, they're referred to as the A2 and P2, the aortic and pulmonary components. And the width of this split, the separation between these components, changes with respiration. So when you inhale, you have a certain spacing between the two components. When you exhale, you have a different spacing between the two components. This is another reason that analyzing these sounds and trying to correlate them becomes so complicated, because if you happen to get one sample from a person while they're inhaling and another sample from the same person when they're exhaling, the signals look different. So here's an example of that. Inspiration, inhale. Expiration, exhale. So this is during the systole cycle. And so we have the S1 and the S2. And then in this case, we have the S1 and the A2 and P2 here are separated. When in exhale, they're closer together. They merge together. Now, why is this relevant? For our cases, it's relevant because there are some diagnostic cues that are relevant when you look specifically at the A2 or the P2, which means that you have to be able to find them. And in cases like this, separating them is essentially impossible. In this case, you have a chance of being able to do it. So physicians listen primarily to 6 locations that I'll show you. And I'll show you a figure that shows that. And so in our case, what we're seeking to do, we call it multisite ossification, is acquire the sounds from all of those 6 sites at the same time so that we can avoid this time alignment issue. Yeah.
Do you also take ambient sound?
We could, but the only use that it would be for us is to try to eliminate it.
Speech?
Yeah, we
Air
don't.
conditioning and stuff.
Yeah, it turns out that it's picked up, but currently we don't try to sample. I mean, you could do noise cancellation, for example, if you did it properly, but at this point we're not doing that. So since we can get them simultaneously, we avoid this propagation time, well, avoid the differences in time alignment. And so within propagation time, since different locations are different distances from the heart, the propagation time of the sound is different between the different locations. But within that difference, these guys that we get are time aligned. Now, by the way, the equipment that we use to do this is technically available on the market. But at the time that we started this, there was no such thing. And so we partnered with a company called Zargus to use their prototype multisite ossification systems so that we could take them into the clinic and gather data using those before they were ever for sale. Zargus doesn't exist anymore. Siemens bought them up, which is a good result for them, actually. They made quite a lot of money that way. But you know, it isn't something you can get off the shelf generally, and they're really, really expensive even if you do get them. So here are the sites that we're looking at. I mentioned the aortic site because it's close to the aorta, pulmonic site, close to the pulmonary artery. There's our 2 side ones that are very far from the heart, called the right and left axilla. There's one that's near the tricuspid valve and another one near the mitral valve. So those are the 6 sites that are standard for physicians to use. So I mentioned that physicians listen to these different locations. And you might say, well, okay, I kind of qualitatively get what you mean because, if the mitral valve is the problem or the tricuspid valve is the problem, listening close to it kind of makes sense. But this is to illustrate how different the signals are at these different locations. So for a particular case, that particular patient, this is what the heart sound signal looks like at the aortic site, this is at the pulmonic site, the tricuspid, the mitral, the right and left axilla. So now you can see if I was to say, well, I'm going to acquire each of these signals individually, and now I'm going to try to time align them by correlation, the signals are nothing alike. And so trying to align them by correlation, they're not correlated really. And so, and we try, believe me, we try. And so acquiring simultaneously is really the only way to be able to use these. If you want to use correlations, you know, if you want to look, well, there's something happening here in the aortic and something happening here in the tricuspid. If you want to be able to do that kind of thing, this is the only way to get signals that you can do it with.
Todd, is it, as far as the equipment, are there like 6 of these stethoscopes that they are put together? I mean, on the
Actually, that's a good question.
Yeah.
It turns out, you can find them on Amazon probably, that there are some really inexpensive adhesive piezoelectric sound sensors that are designed specifically for heart sound acquisition.
Right. Yeah.
Just disposable.
Right.
And they're a buck a piece or something. That's what we use. And so, especially for us when we're dealing with babies, you can imagine, you know, getting them to sit still while you move around 6 different locations. Good luck with that. So you just adhere the 6 piezoelectric sensors, and then,
They are together, and then you get the signal at the
and
same
then,
time.
then we pull cables from those, put them into the acquisition device, and take them from all 6 sensors at the same time.
Together.
So I'm going to talk about a system, and then I'm going to finish up enough so that you can hear about the other half of the problem, because this is just the first half. So I'm going to talk about the signal processing system architecture that we're proposing. And I'll show you how much of it we've actually done and how much of it is yet to be done. So the system that we're going to look at a block diagram of in a second does noise reduction, because you've seen there's a lot of noise. Heart rate estimation, because the heart rate determines the characteristics of the sound. As I said, at high heart rates, things are different than at low heart rates. We extract some features that allow us to determine what parts of the sound correspond to S1s, what are S2s, what are S3s, what are S4s. That the process of using those features to determine which regions correspond to heart sounds is referred to as segmentation. What segmentation tells you is that, okay, there's a heart sound here, or a component of a heart sound here, and another one over here, and another one over here. What it doesn't tell you is that an S1, or is that an S2, or is what? And so that next step is labeling. You have to be able to determine, looking at the segment result, what heart sound those correspond to. It's harder than it might seem, because if you think about it, well, your heart sounds always start with S1. No, they don't. I mean, you acquire the sound from the patient. It may start at S2, it may start at S1, it may start in between, you just don't know. And so you can't say, well, the first one's going to be S1, because it may well not be. And following that, we have to look at symptom detection. That one we haven't really done quite so much with. And we want to do this on the multi-site signals. Once we do that, once we've detected symptoms, then we pass that to the decision support system that we'll hear about in a couple minutes. So here's the architecture we're proposing. It's lots of blocks and lots of arrows. So I'll kind of step through it. So this line through here is what we've done so far, one of my students and I. And so we do the noise reduction. Now it says noise reduction 1. I'll tell you why there's noise reduction 1 and noise reduction 2 in a second. We estimate the heart rate. We extract features that we find important. It happens that for the methods that we've been using, we use the wavelet decomposition to extract the features that we're interested in. Then we apply the segmentation, which is to say we mark places where we believe there to be heart sound components. And once we've done that, then we label those marked areas. Is it an S1, is it an S2, or what? Okay. And then that passes on to symptom detection. Now, one of the things that I haven't talked about here is, well, this murmur stuff that I mentioned before. Murmurs have this really inconvenient feature in terms of analysis, in that they look a lot like noise. In fact, for all intents and purposes, in terms of their spectra, they are noise. And so if you do a really good noise reduction, which makes these things feasible, you remove the murmurs. If your diagnosis requires that you know that there are murmurs there, and many diagnoses do, if you just follow this path, you don't have that information. You can't get that information. And in order to reintroduce that information, I've added, this lower bit here. So what's happening is we have a different noise reduction method here, one that doesn't reduce noise so much, but reduces it a bit that preserves the murmurs in particular. So you can't do much in terms of noise reduction actually and still preserve the murmurs. So this could be a wire for all intents and purposes because you really can't do too much. So here we have this nice clean signal with the murmurs removed. Here we have the signal with the murmurs still there. So we want to keep the murmur, so we got a plus here. We want to get rid of the normal signal because that's dealt with up here. So we have a minus here. And we pass that on to see if we can detect any murmurs. So after we've gone through this process, if there's no energy left, then there were no murmurs. So then we know that. Now we have the heart rate feeding down here because the murmur location and the degree of murmur can vary with heart rate. Okay? So if we've detected murmurs, we're just saying, okay, there are some there. Then we need to say, where are they? Because as I said, the different murmurs can happen at different places and they reflect different pathologies depending upon where they are. And so the next thing we need to do is localize those murmurs. And in order to do that, we need to have the labeled signal, S1, S2, and so forth, because we want to say, well, is it between an S1 and S2? Well, we need to know that there's an S1 and S2 and where it is before we can say that there's a murmur between S1 and S2. So once we've done that, we've found the murmurs if they're there, we've localized them, found their locations, then that's a valid symptom that we can pass on. And so to pass on to decision support, our symptom detection will use the labeled, essentially noise-free signal. It'll use the murmur information that we've gotten, and we'll also potentially use the almost un-noise-reduced signal that we have. That we then need to apply symptom detection to. Yeah?
Is noise reduction just a low-pass filter, or is it more complex?
It can be just a low-pass filter, but generally speaking, one of the things that we need to preserve is these clicks that I talked about. They're very high frequency impulses. And so if you do low-pass filtering, those things just disappear. So it's generally speaking a better approach to use a nonlinear method, like median filtering or something like that, to preserve some of those transients. Okay. So that takes us to the decision support section, which is the next speaker.
I'm working on the decision support system part of the project, and I've been working on this for quite a long time. The decision support, as you would assume, is kind of accepting information from the signal processing. One of the important things that we have to do is separate normal from non-normal cues. And the particular system that I've written does two forms of reasoning that are complementary: recognition-based reasoning and then solution construction and evaluation. And what it produces is not this person has this disease. It gives you a list of possible solutions that it's looked at, and it will rank them according to how many of the non-normal cues they explain. So we'll see an example in a second. So this is the block diagram. You can see we have from signal processing here. It looks at all the cues, separates normal and non-normal ones. Those go into the recognition-based reasoning system. We have a database of diseases to cues, so what is present normally in a particular disease and what cues will lead us to suspect that a disease is here. We have to do it both ways. Once we come out of here, what we have is a list of relevant hypotheses. And those go into the solution construction and evaluation section, and those have a database of cue weights. So maybe some cues, like the click, are more important. So we'll have a higher weight associated molecular cue or lower weight. And the output is ranked solutions and explanations. So here's an example. The person has a small atrial septal defect. Atrial septal defect is a small opening between the upper two chambers, atria, in the heart. And what it means is that blood flows back into the heart and goes around into the lungs. You don't get enough oxygenated blood to the body, depending on how big it is. So in this particular case, we have these cues. And S1 is normal. S2 is split, fixed, and wide. That's not normal. Okay, so this is the normal one that you saw before. We have a split here, and then they're combined here. So it's a variable split. Here we have a fixed split, and it's wider. So that's why it's called a fixed wide split. That's significant. We don't have S3 or S4. That's cool. We do have a murmur, and the time is the systolic phase. So between S1 and S2, it's an ejection murmur, which is in the middle of the systolic area. And it's heard loudest in the pulmonary area. So you have those sites. Loudness is 1/6, and radiation goes to the back of the person. There are no clicks or snaps. So this is kind of what we have to go with. This case is simplified in that I'm only showing you the heart auscultation data. There's a lot of other data that the program can use in reasoning, but this is the main guts of how it works. So recognition-based reasoning, it's going to identify the non-normal cues, and those non-normal cues would then activate hypotheses that include diseases that can produce that non-normal cue. So in the case we're looking at, the S2 being split, fixed, and wide suggests an atrial septal defect. That's the one hole between the upper two chambers. And the other one is a pulmonary stenosis, which is a restriction around the pulmonary valve. The other non-normal feature that we have is a systolic ejection murmur, heard loudest in the pulmonary area, and loudness is 1/6. It's very soft. This suggests you may have an innocent systolic murmur. There are 4 innocent murmurs. They're called innocent because there's not a disease associated with them. They're present in some normal people. Typically in babies, they all have murmurs for a little while. It's just because the distance between their chest and their heart is so small that in a normal person, an adult, the sound is attenuated enough so you don't hear it. So you can't have that in normal babies, normal infants. And so the innocent systolic murmur and atrial septal defect and also pulmonary stenosis. So based on the recognition-based reasoning module, we have 3 potential candidates to look at. And what we do then is we look at what those defects would produce in cues. So we're trying to match up what we have in the case with a potential solution. And it's a little bit tricky because different ones have different numbers of non-normal cues. So we're not going to end up with the same denominator on all of these at the end. So atrial septal defect, this is what it usually produces. Pulmonary stenosis, it has an added systolic ejection click. That we didn't see. Innocent systolic murmur, that has the murmur, but you'll notice that the S2 is normal. It's a variable split, medium width. If we look at a completely normal signal, that has the variable split S2 as well. So S1 is always single. So we look at the differences here. There's some pretty significant differences between some of these in our case. But basically our case looks like this one. So when the second module starts, it has those possible diseases in its memory that have been triggered by the recognition-based reasoning. And there are several settings in the program. You can actually look exhaustively at everything. If you want to look at every possible diagnosis that you can do that. Or the default method on the system is to look at what we have, what's hypothesis by recognition-based reasoning, and if any of those explain everything that we have, we'll give that as an answer. You can go back and look for more things if you'd like. That's the typical way it's done. So we take each of these potential solutions and we evaluate them with a metric I'm calling evidence points. It looks like a really scary equation. No. These are the non-normal cues, and they can be explained with expectations. And so we have the explained ones here over the explained and unexplained ones. So you get 100% if everything is explained, and if whatever's not explained adds to the denominator. So in the formula, if we don't want to introduce the weights at the moment, we just give a 1 in the numerator and a 1 in the denominator for each abnormal cue that is explained by the disease that we're looking at. If it's not explained, then it gets a 0 in the numerator and a 1 in the denominator. If the cues are normal, we ignore them. So if we're looking at our the possibilities that we have for case 212, normal is on the left, and normal doesn't match. Variable split medium doesn't match wide fixed split on the S2, and no murmur doesn't match systolic ejection murmur, no clicks. This does match, so but it's normal. So we get 0, 0. So the total of a normal case gets 0 out of 3. Not good. So then we look at ASD, and that's singles correct, but it's normal. Wide fixed split S2 gets 2 out of 2 for that. In this case, I'm counting 1 for the wide and 1 for the fixed. So you could count it as 1 just for the S2 being different, or you could look at it by the components, because it can be wide, can be fixed, etc. So here I just put down 2 for that. And systolic ejection murmur, it can explain that. You will have a larger number of, I guess, loudness values that are expected from a disease. So ASD, you could expect a, instead of just always being a 1 out of 6, it could be a 1, 2, or 3 out of 6. Excuse me. Click none, so the ASD gets 3 out of 3. Spoiler, that's the right answer. But we also want to look at everything else that has been suggested by recognition-based reasoning. So pulmonary stenosis, we get the same S2, wide fixed split. We look at the systolic ejection murmur, pulmonary area, we get that as well. But if we had a pulmonary stenosis, we would have a systolic ejection click in the pulmonary area. We do not have one, and so we get bumped down 1 here. So we have a total of 3 or 4 here. So that's the way it goes through mechanically all of the different ones. We look at an innocent murmur. The innocent murmur doesn't explain the change in S2, but it does explain the murmur. There's no click, so we have a 1 out of 3 total on that one. So one of my students worked very hard to be able to create this graphical representation of the auscultation PCG that we see at the top. And he also did a more or less complete medical record. There are a lot of other types of information that the cardiologists use to do the diagnosis. This one indicates the radiation to the back of the murmur. This one is the pulses, and they're all 2+. This says the murmur is louder when patient's lying down. It disappears when the patient is standing, and there's a little bit more detail about the S1, S2 here. And these are the details about the particular case here and the murmur. So we have a place for up to 5 murmurs. So it's not unusual to have more than one murmur. It could be one defect causing more than one murmur, or it could be multiple defects causing those murmurs. And because there's a very difficult matching problem going on, and due to space limitations, the maximum number of systolic murmurs is 2, and the maximum number of diastolic murmurs is 2, and continuous murmurs is 1. So we ended up doing that on the medical record here, and if there are no extras here. One of the big problems that occurs when people are trying to diagnose these types of cases is that the complex information that's involved with the murmur, it doesn't matter to us which we call systolic 1 or systolic 2. We could have one early, one late, both mid, whatever. But when you're matching up things, the details tend to get lost. So try to do a neural net. Think of a neural network. Looking at this, it says, oh, it's got a systolic murmur. But you have to have the time and the location and the loudness of it to really be able to match it up. And then if you have 2 systolic murmurs, well, maybe if the neural network expects one first and not the other one, it can't learn. So there's a real problem with some of the traditional ways of diagnosis that are used with this type of information. And one of the other difficulties is that there's no standard format, really, for phonocardiograms. EKGs, been there, done that. PCGs, not so much. So heart auscultation is a widely used low-cost screening method for heart disease. With the exception of expert cardiologists, physicians' auscultation skills are limited, and this includes really seasoned general practitioners, and just about everybody else includes cardiology residents for a time. There have been many standard training methods applied to this problem, and I could talk about some of those if you're interested, but they haven't been able to give an improvement. Sometimes there's an improvement for a couple of weeks or a couple months, but people can't, if you don't hear the sound all the time, you just lose it. So what we're proposing is this analysis system so that we can analyze the signals and then pass them to decision support and give some indication to a physician what we think is going on with the patient. We're not going to say they've got atrial septal defect, even though we might know that in the case of our example. But we present what patient has and what kinds of defects explain all of those abnormal cues. And so it's up to the physician to do something with that information. There's a couple of acknowledgements. Yan-Han Ni was the student that wrote the graphical information, the graphical representation of the patient data. We had a grant from the Telemedicine and Advanced Technology Research Center Army Advanced Medical Technology Initiative. And Dr. Mankey helped us immensely with, primarily by providing the phonocardiograms and having the multi-site setup, which is not very common. And we talked quite a lot with him. And he's with Tripler Army Medical Center, which is in Honolulu, Hawaii.
Okay. I'm interested to know what sort of, how you can go about to identify some features, to go and then write algorithm and so on to come up with automatic type of recognition and stuff. Now, let's say, for example, in the case of the bicuspid aortic valve, then you do not have three, and then you have to. And then basically the fundamental frequency of the heartbeat will be the same, whereas it's the the extra maybe frequency or whatever they come in, and then when the blood is pushed against the aorta at the top of the arch, and then it makes in fact dilation and all those things. Now, how do you go about identifying a feature for this?
Well, the way that we're doing it is using wavelet decompositions. And there are a number of different ways, but the main thing that you need is something that extracts features in a time-sensitive way. So if you say, well, you've got this whole signal, and I'm going to find a feature for this whole signal, that's meaningless because there are specific points in time in the heart sound that are meaningful, and their characteristics and their meaningfulness changes with time. And so you need some sort of analysis method that accommodates time. So Fourier analysis, forget it. You need something that accommodates the time change. Wavelet decompositions do that. They're called time scale decompositions generally. Time frequency analysis does that as well. And so either of those two methods are interesting. The wavelet decomposition is good for features that can vary in their time extent. Time frequency analysis is better for ones that don't. And so we've chosen wavelets, but we haven't exhausted the possibilities for time frequency analysis at this point. But that's the class of techniques we've been using to generate features.
Right. Because there are some memories there in the case of the aortic stenosis and so on, which in fact can be used, but I didn't know how to do that. And then the second question to that is it that you also brought in some samples from the patients? I mean, and then basically look at it and so on, and then see how it goes with the normal, and then to go and then identify the features.
Well, we actually have a database. At this point, we're working with about 50 patient sets. There are many more, but 50 was quite enough to be dealing with. And so, in fact, we can visually compare, or by the basis of features, compare what normal versus abnormal appears to be. We didn't take that approach, but one could extract features and then do training in order to compare these things. If you try to do it visually over a large set of data, it's helpless.
Yeah.
But I mean, it's possible to use like a base normal and to use that to look differentially to determine pathology. But what we've been seeking to do is just find the signature of the pathology without regard to a normal. Because again, between patients, comparing their signals is really hard. Because even several normal patients have very different heart signals. And so trying to find a base is difficult. And so we've been seeking just to find the features that indicate pathology regardless of what other features of the patient are.
Yes. I was curious, do you know what software your student used to create that?
Java. Yes.
Assuming you have 6 or whatever number of piezoelectric sensors connected to something, what is the hardware involved before the computer? I mean, between the computer and the patient. What else is there?
Okay, so we have these 6 sensors. Each of them go into independent channels. And so since the signal coming off the sensor is analog, the first thing to do is an A to D conversion. And then we have to choose a quantization level for the A to D conversion. But basically, I mean, it's large, it's modular, it's very high quality instrumentation quality A to D converters. But basically that's it.
But you didn't make them, they are.
No, we partnered with this company, Zargus, that was doing prototypes
Ah,
of a system like this, and got their they lent us their prototypes to work with. And they've since been FDA approved, and, well, actually they were FDA approved because we couldn't put them in clinic if they weren't. But that's where they came from, because we could technically design them, but it would have taken us 2 or 3 years, and that wasn't the point of the exercise. And these guys at Zargus are really very good.
What's the sampling rate on the A to D?
It was, if I remember right, about 20 kilohertz. Much higher than it actually needs to be, but it's about 20 kilohertz.
Are there other questions?
How'd you get into this in the first place?
That's an interesting question. You want to answer it first?
That's actually a long story. I was working on decision support for pediatric cardiology, congenital heart defects, way back in my PhD days. That was my topic for my PhD dissertation. I didn't do any signal processing then. We had written medical records that we transcribed and were able to put into a computer program, which is written in Lisp, so it's really easy to put all those different characteristics with murmurs and match them up appropriately. And so that's what I did for my PhD, 1995. And since he does signal processing.
In my case, I started out in signal processing with an interest in audio. I since moved into image processing and computer vision and a lot of other things, but I remained interested in audio. And so we came to this point where we found this cardiologist that was interested in working with us, and finding doctors that were willing to work with engineers is not so very easy. And so we came across this guy. And I was starting to develop an interest in biomedical engineering generally, and in particular biosignal analysis. So I was introducing a course in biosignal analysis in Hawaii, which is currently being run at the graduate level. And so that seemed to mesh well with this. And so I had a really good PhD student a few years back that was interested in this type of thing. And so that's really where we started to get involved. So at the point she had the written medical records, we said, well, why use written medical records? We have such good instrumentation now, can't we just generate this stuff instead of having somebody write it down? And so that's where the inspiration came from.
And then are you working more for the grown-ups or the babies as far as getting the data?
Well, in terms of our data access, our contact is pediatric cardiologist, so he has access to babies. And so, I mean, the hope is that the techniques are not baby specific, but I mean, that's where we can get the data. And so that's pretty much driven what our demographic is. But the techniques we expect to be generalizable. The symptom detection and these types of symptoms are likely to be different, but the general approach we expect to be applicable.
What sort of differences do you see between the babies and then the grown-ups?
The incidence of murmurs is different. Babies, as Nancy mentioned, almost all have murmurs. They're almost always innocent murmurs, but not always. In adults, murmurs are relatively rare. They're almost always indicative of pathology. In terms of things like valvular clicks and things like that, they're much more common in adults than in children. And so, statistically, they're different. The symptoms themselves are qualitatively the same because the general system is the same.
The heart rate in infants is very high.
Yes.
And that makes the time between S1, S2, and then S2 and the following S1 pretty much equal. So you can't use that distinction, the length of that, as a clue to which one's S1 and which one's S2. So, and it's so fast. The Army was funding it because they also have people that have very high heart rates under certain circumstances. So they're interested in, in general, but also if there's some way that they can help the soldiers in the field.
The fact that you're working with the telemedicine group, does that mean you send the signals remotely to be processed elsewhere?
Dr. Mankey did a bunch of work, and they did a verification study of physicians diagnosing the diseases and interpreting the murmurs via telemedicine. So Tripler is responsible for all of the Pacific Islands. So there's people in Guam, all over, and it's a pretty expensive plane ride for the physician or for the infant to make that. If the general physician finds a murmur and doesn't know if it's pathologic or not, it's estimated to be about $5,000 to
The
get
main
that.
equipment is with the patient.
The acquisition is with the patient.
Okay.
And then they have a system where they can access those sounds from here. So before they had the system set up, they had to fly, or the patient had to fly to Hawaii.
But the processing that we do is local. So we have the sounds locally, we have the processing locally. We're not using a telemedicine link at this point. Although the graphical representation that was shown there is used in their telemedicine.
But you could send the pulses
Sure.
remotely to some
Yeah,
other center,
yeah.
wherever they.
Yeah, yeah, could well do that.
MP3 file.
No, no. MP3 files are highly compressed, which ruins the statistics of the data badly. So, no, we need it uncompressed. But it could be an uncompressed audio file, sure.
And it concentrates on voice, the MP3. So the frequencies that you need for this are not present anymore.
Okay, now the PISA is here, and I understand that Todd has a class at 6 o'clock. I would like to thank them both for giving us such an interesting and instructive. In fact, for me, it was really instructive, and I bet you have also learned something out of it. So please join me in